Yeah, so I moved to Lethbridge from Vancouver about 5 years ago, and I think I said in my bio, I'm still waiting for those nice snowy winters, because I thought I'd be able to cross-country ski out my backyard to work every day, moving from the west coast to Alberta, but that hasn't happened yet. So I'm keeping my fingers crossed for more snow. Anyway, so today I'm going to talk to you about Aphanomyces root rot, but we can't talk about Aphanomyces root rot without also talking about Fusarium. So I'll talk about those two pathogens today. So just an overview of what I'm going to talk about today. So I'll start with talking about what the causal agents are here in the prairies. What are some of those risk factors for causing root rot? What are the host range of our root rot pathogens? How are they distributed in Alberta? And then I'll wrap up by talking about some of our current research projects that I'm doing now. Okay, so we have two main pathogen groups of focus. The first one is Aphanomyces euteiches, and I like to call this one our A-list celebrity pathogen. It gets all the attention, whereas Fusarium species I usually call our D-list celebrity pathogen. It's kind of like the Kardashians. You don't know why they're famous. They're everywhere. Supposedly they do something, and so we're trying to figure out what it is that they do. Okay, so I'll start by talking about Aphanomyces euteiches. The first report of this pathogen was in 2013 in Alberta, and just because Saskatchewan always has to do everything a little bit better than us, they found it first in 2012. And we thought, oh, if Saskatchewan has it, we must have it too. So we looked for it in Alberta, and indeed we did. This is a pathogen that's called an oomycete, or a water mold. And that's because it has a portion of its life cycle that you can see here. It's called the zoospore. It's got these two little flagella, single-celled, and these allow it to swim around in free water. So because of that, it's called a water mold. The other part of its life cycle is here. It's called the oospore, and this is a very long-lived resting spore. If you look closely, you can see that it has this very thick cell wall, and that allows it to withstand lots of differences in temperature. So particularly when we're looking at survival in the soil, and we haven't had those really cold winters in the last 5 years, we're not getting those really extreme environmental conditions that would maybe help to degrade this resting spore a lot faster. Currently, there's only one registered chemical against this pathogen, and that is called ethoboxam. Trade name is Soll, but it's only registered for early season suppression of this disease. And it was registered on all pulse crops in 2015, except for peas. So in 2015 and 2016, there was emergency use granted for this chemical on field peas, and I think they're pursuing full registration for peas. The biggest problem with this pathogen is that it's very difficult to isolate as a plant pathologist for us to determine that that was the causal agent. It wasn't until we moved into using a DNA test that we actually found that, lo and behold, we have it in Saskatchewan, we have it in Alberta, and we have it pretty much anywhere peas are grown. Okay, so here's the life cycle, just to give you some sort of history or give you a clue on its biology. So as I mentioned here or earlier, it always starts as oospores, these thick-walled single cells that are produced in the host tissue, and those are left behind in the soil as those root tissues decay. Once you plant a susceptible host, and that's the key here, that susceptible host releases chemical signals that induce these oospores to germinate. They go through kind of this complicated germination cycle to eventually produce the zoospores, so those little single-celled organisms that can then swim to the roots. They're really good at finding their host roots. They quickly attach and then start to spread throughout the whole root system. And they can infect a root in about 7 to 10 days. As those nutrients get used up and everything starts to degrade, then they go through sexual reproduction to then again produce those oospores that are left behind in the roots. So that is the sort of life cycle for Aphanomyces. If we look at the symptoms that it causes, these are very typical symptoms where you get complete decay of the roots. browning of the lateral roots here, pinching of the epicaudal that stops very abruptly at that green tissue. In more advanced stages of the disease, if you pull up the pea plant, often all that you're left with is this little sort of stub of a root, and you'll get a long string of the vascular system that's left behind, but pretty much the whole rest of the root is decayed. And then if you take a look at that under the microscope, what we see here is it's just the roots are filled with oospores, which are left behind in the soil. Here's a picture of a pea field from more central Alberta, close to the Drumheller area in 2014, where we really saw that this was the first year where this disease seemed to really make it showing, and we saw it a lot in that sort of central Alberta area. And this picture is particularly striking because of a couple things. First, down here, you can see this is the low spot of the field. So you get a lot of water collecting down here. You can see we have a lot of the loss of the pea biomass here. And then this area of the field here was nice and green, and that's because of the crop history of this field, where this portion of the field had been in pea production for about 20 years, crop rotation, peas one in every 3 to 4 years. And this portion of the field was the first year that had actually been planted to peas. So you can see just what the difference is on a long crop history of peas versus first year pea crop. And then you can see, however, that just by seeding operations, by sort of moving that soil back and forth during seeding, that you are starting to move some of that inoculum into the healthy part of the field. Okay, so now we'll move over to our D-list celebrity, our Fusarium root rot. And Fusarium is a very complicated species, or very complicated group, and that's because it's a very large genus that actually has hundreds of different species. And Fusarium as a group of fungi is one of the most common fungi in the soil. Most of them are actually there to help degrade that organic matter, to help break down nutrients in the soil. But there's a few of them, of the species, that are plant pathogens. And probably one of the most well-known Fusarium plant pathogens is Fusarium graminearum, which causes Fusarium head blight. So here I just want to stress that the Fusarium that causes Fusarium head blight is not equal to the Fusarium that causes Fusarium root rot. The species that we're primarily dealing with is Fusarium avanaceum, but there's actually a number of different Fusarium species that are involved, and I'll show you what some of those are in the next few slides. So when we look at the symptoms of Fusarium root rot, what we see often happens is it starts right at this point of seed attachment. That's where you get a lot of nutrients leaking out of the seeds. Fusarium is a pretty weak pathogen, so it's going to take advantage of that. It starts to spread along the taproot. You get lesions spreading along the taproot. Eventually, you'll see they cover the whole taproot, and you can eventually get your plants, your roots looking like this, just sort of a blackened mass that's left of the taproot. If you cut that open, oftentimes you'll see this really bright red streaking in the vascular system, which is a really good indicator that it's Fusarium. Okay, so how do we distinguish Aphanomyces versus Fusarium? Well, in the lab in the greenhouse where I inoculate plants with Aphanomyces and Fusarium, I can tell them apart very easily. So I'll show you how I do that, and then I'll show you what roots actually look like from the field. So Fusarium, we get this blackened taproot that starts at that point of seed attachment. Most of the time, the lateral roots will still look pretty healthy, and because of that, we still usually see nice, healthy shoots. In comparison, Aphanomyces, we get that pinching of the epicaudal here that stops very abruptly at the soil line. Oftentimes, we get decay of the lateral roots that look soft rot, a honey brown decay. And because we're getting the whole root system being affected, we very quickly see this yellowing and wilting of the shoots. So in the field, if you're seeing plants that have this very characteristic yellowing and stunting of the shoots, it's a good indicator that you're dealing with Aphanomyces, whereas oftentimes we see that plants in the field that have Fusarium root rot can still look fairly healthy. Having said that, when we go to the field and we start collecting roots, the symptom expression is really not clear-cut. So it's really hard if you've just dug up some roots and are trying to figure out, do I have an Aphanomyces problem? Do I have a Fusarium problem? Do I have both? It's actually really difficult to tell just by looking at the roots. And actually what most often is the case is that the roots are infected with a pathogen complex. So usually if we find Aphanomyces, we're finding Fusarium at the same time. Okay, and in terms of some of the differences in these two diseases, Aphanomyces root rot is, I guess, the A-list celebrity because infection can occur at any time in the growing season. Most of these root rot pathogens, the most susceptible stage of a plant is around seeding, or sort of that 4 to 5 weeks after seeding. Whereas Aphanomyces, we find that the most critical period for infection is if there's soil, if there's moisture present in that soil. So if you have a nice healthy pea plant, you get a bucket of rain like we have been lately in July, as soon as there's some excess moisture in that soil, Aphanomyces can infect. For Fusarium root rot, we find that the most susceptible stage to infections seems to be at that seedling stage, and then the disease slowly compounds from seedling stage up to flowering, and that's often times when we don't see symptoms appear until flowering. And we've also found that peas can sustain a fair amount of Fusarium root rot before the yield is impacted. So here's an example of some field trials that we looked at. And we have a site in Lethbridge and we have a site in Tabor. And our site in Lethbridge has Fusarium only, whereas our site in Tabor has both Fusarium and Aphanomyces. So what we do is when we collect these plants, we rate them for disease, and we rate them on both a Fusarium scale and an Aphanomyces scale. And then here I've just collected all that data and looked at, just combined it to see what the yield was. So you can see here when we only have Fusarium, we actually had fairly good yields in this trial, kind of ranging from about 2,000 up to 4,000, just depending on which treatments we were looking at. But if we compare that to where we had Aphanomyces present, our yields are down to about 1,000 kilograms per hectare and only up to about 3,000 kilograms per hectare. So we're seeing a fairly large yield penalty from Aphanomyces that we don't often see if there's only Fusarium present. Okay, so what are some of the risk factors for root rot? Well, the number one risk factor is crop history. And that is particularly once you've had a field that has had about 4 to 5 cropping cycles with a susceptible host. And for the most part, we're talking about peas in Alberta as kind of a cropping history, but when we look in Saskatchewan, you have to take into account lentil production as well. The next most important risk factor is soil moisture, and that's particularly for Aphanomyces. And this is because the oospores can germinate in dry soil. So if we think in 2016 where we had very dry weather in May, those oospores can still germinate in the presence of the peas, but they're just gonna sit there until there's enough moisture. As soon as you get rainfall, they hatch out those zoospores, and then the zoospores will swim to the roots. And then the other, some of the soil characteristics or other important risk factors, one of the most important ones being soil compaction. So it's really, the really conducive soils are ones that have fairly high clay content. And so that would be up in that sort of Drumheller to Three Hills region where they have the really heavy gumbo clay soils is where we can often see that root rots will be the worst. Soil pH, more acidic soils are more conducive to disease. And soil temperatures, the optimum temperature is about 24 degrees. So that's often why we don't see disease develop until sort of late June, early July when our soil temperatures have really warmed up. And the presence of other soil-borne pathogens. So not only Aphanomyces, we have Fusarium as well, other soil-borne pathogens like Pythium and Rhizoctonia that are all part of this complex. So it's quite a complex system that is causing this disease problem. Okay, so we conducted surveys for pea root rot over the past 4 years, I think, yeah. And what we did, when we started out in 2013, we actually didn't know what was causing the problem. And we thought, well, let's get away from culturing our roots. Generally, plant pathologists, you collect your samples, take them back to the lab, plate them out on a media, see what you get. We already knew that if we just did that, all we were going to pick up was Fusarium. So we moved into DNA testing, and that's really where we could start to see what the differences were in some of these pathogens. Okay, just for context, this is a disease severity rating scale that we used. So 1 is healthy here. Plants have a lot of nice pink nodules. That's what we want to see. And then moving all the way up to 7 where the plants are, the roots are completely dead. Okay, so what did we see over the past 3 years? And I'm only showing data from 2014 to 2016 because we didn't know what we were doing in 2013. So it took us kind of a year to figure out that the protocol. So I'm showing the data from then. And I've broken it up from different soil zones, from black, dark brown, brown, Peace River. It's harder to get samples up from Peace River, so we didn't get any from 2016. But we can see in terms of severity here, where we're moving into the plants being, moving out of that healthy zone, and most of the plants having a moderate root rot. And in 2014, it was worse, the root rot was worse in the dark brown soil zone at 3.9. 2015, the plants actually looked a lot better, and that's because it was a very dry year. And there actually was roots from the brown soil zone that had the highest disease severity at 3.4. And then 2016, we didn't know what to expect. Was it a dry year? It started out being a dry year, and then it ended up being a wet year. So our severity has kind of fallen somewhere in the middle there. But again, we saw the highest severities in the brown soil zone. And then we also looked at percent of the fields that were positive for Aphanomyces from 2014 to 2016. 2014, very, very wet year. We saw the highest percentage of fields positive for Aphanomyces in the black soil zone at 83%, followed by the dark brown soil zone at 56%. And then the brown soil zone was actually pretty low at 34%. And in the Peace, we didn't actually find any fields that were positive. So we thought, well, that's good. It at least hasn't reached up that far north. But then in 2015, which was a really dry year, you can see here that there was a lot less fields testing positive for Aphanomyces, but we did find it in 50% of the fields that we tested in the Peace River, indicating that it had reached that far north as well. And then again, like I said, in 2016, we really didn't know what to expect with kind of the wacky weather, and we actually saw that we had fairly high levels of fields positive for Aphanomyces. And surprisingly, it was at 56% down here in the brown soil zone, which was the highest we had seen yet out of the years that we had tested. We also found that Fusarium species were present in all the fields that we tested, which again just really makes this kind of a really complex disease to deal with. And when we look at the Fusarium species that are present, this is why it's challenging to work with this group of organisms. We test for about 10 to 12 Fusarium species, and we can usually find all of them when you start looking. And so we find a lot of two species here. Aphanomyces and Sollani are very high in all the fields. Soll, redolins, and oxysporum, so these are just different species. And when you start looking at what are the virulents, we find that actually Aphanomyces and Sollani are the two most virulent pathogens, and they're present in close to 70 to 80% of all the fields that we're testing. These two species, redolins and oxysporum, are very weakly virulent, but again, very, very commonly distributed in all the fields. And then some of the other ones, like Calmorum and Graminearum, are more of a wheat problem, and we don't really find them to a great extent in peas. So this Fusarium species complex is very challenging to work with and very challenging to try and figure out which are the ones that are actually causing the disease. Okay, so the next thing we looked at was host range testing of Aphanomyces. And we did this in the greenhouse and in conjunction with Sabina Benitza at the University of Saskatchewan. And really what we found is that peas and lentils are just as equally susceptible to Aphanomyces. You get lots of oospores being produced in the roots, and that means that every time you're growing these crops in a field, you're increasing that oospore level, increasing your inoculum potential. And then we also tested some of the forage species, and Sicer milkfetch is also very susceptible to Aphanomyces. Dry bean and alfalfa give very variable results, so some cultivars are susceptible, some cultivars are resistant. But in terms of dry beans, the roots actually stand up very well to infection by Aphanomyces, and we get very few oospores being produced, meaning that they're not going to help increase that inoculum potential. Alfalfa is really dependent on the cultivar tested. And then pulse crops that are resistant or would be considered non-hosts are chickpeas, faba bean, and soybean, and then sainfoin and fenugreek as forage legumes are also very resistant. So these are good options to plant if you have an Aphanomyces problem. So we wanted to go out and see, okay, we can look at this host range in the greenhouse, what's happening out in the field. So in 2015 and 2016, we went out and surveyed all the other pulse crops. and alfalfa is our forage crop. And really what we can see is that alfalfa, dry beans, and faba beans, for the most part, really do not have a root rot issue. Very low incidence and severity in alfalfa and dry bean. Faba beans had some root rot issues in the brown soil zone, and that's more of a Fusarium root rot problem than an Aphanomyces problem. But lentils here, if we look at the incidence and prevalence, is as high as we see in peas, but the severity is a little bit lower. And if we go and look at what are the root rot pathogens in lentils, and here it's a comparison between Alberta and Saskatchewan. So I guess this is maybe one instance where we're happy Saskatchewan is beating us. We can see that they have about a 45% positive field. 45% of lentil fields are positive for Aphanomyces, whereas in Alberta we had about 25 to 30% of fields that were positive. But those Fusarium species are coming back to rear their ugly heads again, where we can see we're looking at about 70 to 80% of fields that are also of lentil fields that are also positive for Fusarium. Okay, and in terms of root rot symptoms, peas versus lentils, really you can see that there's not a lot of difference there. You see that same sort of decay of the lateral roots and the browning of the epicaudal. I think the big difference here is that lentil roots are a lot woodier, so they do seem to withstand a little more damage than peas can, whereas the pea roots are really kind of soft and fleshy, and they degrade very quickly. Okay, so for control options, I guess the big question that I always get is, what can we do right now? And really, the number one recommendation is that if you've had a history of root rots or you're worried about root rots, then you should get your soil tested to confirm presence of Aphanomyces. So most of the seed testing labs, BioVision, 2020, Discovery Seed Labs, BDS, they all offer an Aphanomyces test, but right now they can only confirm presence or absence. And then if you know that you have Aphanomyces, then you should avoid planting peas or lentils in those Aphanomyces infested fields and prolong your rotation of susceptible hosts in those infested fields for about 6 to 8 years. And so you can consider planting one of those other resistant pulse crops like faba bean, chickpeas, or dry beans if you're in an area where you can grow those. You can consider using a seed treatment that targets the root rot complex, and this is particularly more effective against Fusarium species. I think with the early season suppression of Aphanomyces, you'll get good early season control, but that might not relate into a long full season control. And I just want to kind of stress that an in-crop fungicide application unfortunately doesn't have any effect on a root rot. There's no fungicide chemicals that you can spray on the shoot that move down into the root and that will help control the disease. Okay, so this is a fairly new problem or new disease for Alberta. So we're still in a fairly heavy research phase for this disease. And right now, my lab, we're working on about 5 different research projects. If I went through all of these research projects, I'd probably be way over time, and I see I don't even have much time to wrap up what I wanted to. So today I'm only going to talk about what we've been doing on trying to develop inoculum quantification tools. We've been doing a lot of field trials for the past 2 years, but I don't have all the data together from our 10 site years to be able to confidently show you those results yet. Hopefully they'll be coming by next year or the next few months. Okay, so what we've really been actively working on is trying to develop predicting root rot risk. So similar to what Susie talked about earlier about predicting sclerotinia risk, I think one of the big things that we can do is try and develop a tool to predict root rot risk in the soil. And so the idea is that you can take a soil test, you can quantify the amount of inoculum that you have present in your soil, and know if you have a low, medium, or high risk of getting root rot. So there's quite a number of steps that need to go into producing that. We need to make an inoculum dose response curve, so how much inoculum causes how much disease. Then we need to develop a way to test each of these pathogens using DNA tests, and then we have to be able to relate that inoculum level to the DNA test to disease severity. So we're looking at a lot of different steps, and really the step that we're working on right now is trying to understand how much inoculum causes how much disease. So the question that we have for Aphanomyces is how many oospores per gram of soil is going to cause disease? Is there a difference when we're seeing that the pathogen is spread throughout all of Alberta and Saskatchewan? Is there a difference in this inoculum potential between soil zones? Do we have to consider that Fusarium might also be contributing to disease? So what we do is a series of greenhouse tests where we have summer students that spend a lot of time doing nothing but washing and rating roots for months on end. So this is all the roots set out to be rated. And then we rate them on a scale here of 1 being healthy, 3 for Aphanomyces means fairly significant disease, and that's what we're looking at as kind of our threshold level of disease, and then 4, and then 5 is a completely dead plant. And these are really nice tests to do because they're very visually appealing when you look at the results. So here we can see our healthy plants here, and then it's like you kind of flipped a switch, you get to a certain amount of oospores, and everything else is diseased. So we'll take a look at what those numbers are for the different soil zones. So for the black soil zone, these are samples or soils that were taken actually from Saskatchewan in their black soil zone. The first thing that we look at is what happens in the soil if we autoclave it. So there's nothing, everything's killed in the soil, and then we add Aphanomyces back to that soil to see how much we need to cause disease. So here we're looking at oospores along the x-axis here, disease severity, and you can see that there's a nice linear relationship. And then we compare that with sort of raw soil. It's got its full complement of microbes. How many oospores does it take? So we can see for the black soil that we get significant disease developing here at 3 at about between 100 to about 500 oospores per gram of soil. And for the black soils, it doesn't seem to matter whether you're looking at autoclave soils or soils that have that full microbial complement. Okay, for the dark brown soils, again, we have our autoclaved, a nice linear relationship compared to the non-autoclaved raw soils, very same relationship. Here we get significant disease occurring at about 100 oospores per gram of soil. And then for the brown soil zone, again, we have our autoclaved soils. We see that nice linear relationship. And what was interesting here is that you take your non-autoclaved soil, so raw soil, lots of different microorganisms, and we can see that we get a very different pattern between disease. And so for our autoclave soil, where there's nothing but Aphanomyces, we need about 750 oospores per gram of soil to cause disease. But for our raw soils, where we have Fusarium also present, we only need about 100 oospores per gram of soil to cause disease. So it's really significant for the brown soil zones where we have a lot of Fusarium present. There does seem to be an interaction between Fusarium and Aphanomyces that cause more disease. And I'll show you some pictures of what's going on. So here in our autoclaved soils from the brown soil zone, you can see this is our control, so there's no inoculum added. Very nice, healthy, white roots. We've killed everything in that soil. We have nothing to cause disease. Here is natural soil without any added Aphanomyces inoculum, and you can see that we're getting that Fusarium presence already. So you have some Fusarium already present in the soil, you add Aphanomyces, disease gets significantly worse. Okay, so what we found is that there is a variable dose-response curve between soil zones. Dark brown seems to be the most conducive to disease, followed by brown, followed by black. You need about 100 oospores per gram of soil in the dark brown soil zones to cause severe disease, whereas in the brown and the black, you need about 750 oospores per gram of soil. However, if you add Fusarium into the mix, particularly in those brown soil zones, we see that disease severity is increasing to about 100 oospores per gram of soil in all soil types to cause disease. So the next thing that we wanted to do was map where we were finding this Aphanomyces inoculum. So we went out and sampled soil from a whole series of fields. Again, this is in Saskatchewan, and looked at how much, what the inoculum potential was in the 0 to 20 soil layer, 20 to 40, and then the 40 to 60. And what we found here, based on the disease severity ratings, is that really the most of the inoculum is occurring in that 0 to 20 centimeter layer. In highly infested fields, like this one here, you can see that you have significant inoculant potential all the way down to that 60 centimeter layer. In other fields, we see that we still have sort of healthy soils down below. And then we also wanted to map the fields and say, where can we find this across a field? So we just started out sequentially sampling and looked at where that inoculum was spread throughout the field. And really, you can see that we're looking at pretty uniform distribution of Aphanomyces throughout the field. There's some spots that are a little bit lower, but this spot here, 11, was supposed to be kind of our healthy site that we had worked with the grower to figure out where have you had better yields in this field, and we consider that a healthy site. And you can see that for the most part, the healthy sites are often just as diseased as had as much inoculant potential as our disease sites. And this is just another example of another field where you can see again that you have pretty much uniform distribution of the pathogen throughout the field. And this is my plug for, I guess, why all plant pathologists, or maybe all producers, need a drone. I think when we're trying to look at mapping these fields or figuring out where do I need to soil sample to figure out what's going on with my field, if you can get that aerial view of what's going on. So here we have a field that had very patchy distribution, where we can see that you're getting more inoculum along maybe the waterways, lower areas of the field. And this is another field where it had completely uniform distribution. So it gives you a clue of where you would maybe need to do some soil sampling for this. So in conclusion, I'd like to say that root rots are unfortunately fairly widespread, but we do see that they have less of an impact in dry years. Aphanomyces and Fusarium root rots occur as a complex and can be very difficult to distinguish. Before 2016, I thought that in the brown soil zone, we were just dealing with Fusarium root rot, but 2016 completely changed that hypothesis, as we saw a lot of Aphanomyces root rot down here in southern Alberta. Long rotations between susceptible pulse crops are currently our only control option, and faba beans, soybeans, and chickpeas are your best pulse crop option. And inoculant potential differs between soil types. So we see that the dark brown soil zones are dark brown soil are the most conducive to this disease. And I think that reflects what we see in the field, where we really saw the hot spot for root rots, kind of in our dark brown soil zones up in that Drumheller to Three Hills area. The presence of other pathogens, especially Fusarium species, sort of complicates the whole issue. We do see that the threshold dose for Aphanomyces root rot is about 100 oospores per gram of soil, and that those levels are highest in the top 20 centimeters of the field, and we see them decrease with increasing soil depth. And the horizontal distribution that we're seeing in a field can be fairly uniform, but some fields it's still patchy. So with that, I'd like to thank all the funders of this project, and I'm happy to take any questions. Yes and no. So the question was, is anyone doing any research on looking at sort of the whole biology of soils to see if there's any good microorganisms in the soil that will help degrade those oospores? So yes, because this is a really new disease issue, there's a lot of research that's just starting to ramp up. And I don't know if Yantai is still here, because my understanding is that they were going to maybe start doing some research on that at Swift Current, looking at what is the sort of microbiome of the soils, and maybe some healthy versus diseased soils, and see if they can pinpoint what any of those healthy microorganisms are. And then there's also a researcher at the University of Saskatchewan, Fran Wally, who's looking at whether they can isolate any beneficial endophytes or bacteria that maybe will have have a suppressive effect against root rot. Yeah. Yeah, so right now, all varieties of peas and lentils are susceptible. The U.S. and France have been doing research into developing resistant germplasm for the past 20 years, and they do have some resistant germplasm lines that I think can be released for breeding purposes. So I think my understanding is that Canadian breeding programs are going to start looking at trying to move that resistant germplasm into our Canadian adapted cultivars. But breeding, unfortunately, can take a while to get there.
Okay, I think that's it.